All right, so we're going to do a little review on chapter 24 and 26 in preparation for next week's quiz. So next week is week 8 quiz. Sorry, this is week 8 right now, but we're covering chapter 24 and 26. I do have a review that is in your announcement. So make sure that you go over that review again, in addition to this review. So this review is being recorded and it will be uploaded and made available to you, okay? So remember that our immune system is, and chapter 24 is where we're gonna talk a lot about white blood cells, all right? So our white blood cells are going to include, and if you remember back from chapter 16, we talked a little bit about cells, about white blood cells, and these white blood cells are going to be discussed more thoroughly in this chapter. Okay, so if you remember, we have 5 white blood cells in order of abundance, which means the most abundant, the most number of white blood cells that we have are found in our neutrophils, and the least abundant are our basophils. So there's a good way to remember them. It's never let monkeys eat bananas, and the first letter of each one of those is going to be the letter of the white blood cell, and in the order. So for example, never is N for neutrophil, let is L for lymphocytes, and then you have monkeys, M, monocytes, E, eat, that is eosinophils, and then B for basophils, the word bananas, right? So never let monkeys eat bananas helps you remember them in order. Now let me go ahead and talk about them in a slightly different order, because white blood cells are going to be mostly separated by their appearance. So we have the granulocytes, which are the ones that have the multiple lobed nucleus with the granules in them. So the granulocytes are the neutrophils, basophils, and eosinophils. So the neutrophils are the most abundant of all of your white blood cells. They are going to kill bacteria. So whenever you have a high neutrophil count, usually means that you're fighting off a bacterial infection. You could have a low white blood cell count, a neutrophil count, if you've gotten over a bacterial infection. All right, so neutrophils are going to be most important for those. They are part of the mononuclear phagocyte system. All right, so they are cells that are going to consume other things, right? So that's kind of the one part of, okay, so they are not part of the mononuclear phagocyte system, but they are cells that kill other cells. So I apologize, they are not part of that system. I really apologize for that. So they are actually cells that kill other cells by releasing what's in those little granules called granzymes. Okay, so granzymes, they actually poke holes, they perforate other cells. Okay, your neutrophils, again, that's their main function. Neutrophils are going to be really important again in fighting off bacterial infections. Then you have your, let's go to the eosinophils. So the eosinophils mainly are going to function in your, they're going to kill parasites, but they also are part of the allergic response. So they are anti-parasitic, but again, if there are no parasites to fight, and that's part of why we have a lot of, in the industrialized world, we have a lot of allergies, is because we are very hygienic and we no longer have a lot of tapeworms or roundworms. Like, people don't have them in their intestines. So since they don't have them, they are finding that they have an increase in the allergic response. So they are going to basically promote the production, right, of they're going to promote the production of your histamine, which is one way of a response, and also something called mediators of IgE, which is a type of immunoglobulin or an antibody. And that antibody mediates anaphylaxis. Okay, so anaphylaxis is a that causes anaphylactic shock. Okay, so that is part of your functions of your eosinophils. And then your basophils, they have really, the eosinophils, if you remember, they're the ones with the red orange granules, but the basophils have really dark granules. And because they are going to be very, very dark and have lots and lots of granules, they're gonna, in their granules, they release something called histamine and heparin. So they're also going to promote a little bit of that histamine response, which could cause, histamine is a vasodilator, but also a bronchoconstrictor, which means it narrows the passageways of your bronchioles, but it also is going to widen your blood vessels. So they really are going to be there to promote blood flow, and along with heparin, which is an anticoagulant, again, promoting blood flow. Okay, and so they are going to be, so those are your granulocytes, the neutrophils, eosinophils, basophils, make sure you know those. Then you have the mononuclear phagocyte system. I'm sorry, don't know where my brain went there for a minute, is the macrophages, okay? And macrophages are related to a cell called a monocyte. So your white blood cell is the monocyte, but once the monocyte kind of gets to a tissue and it moves its way into the tissue through the capillary and the capillary wall, it's going to become a macrophage inside of the tissue, okay? So monocytes are the precursors of macrophages. Monocytes are very interesting. They are non-granular. They don't have granules, but what they are is they're very large cells. And these cells are what we call antigen presenting cells. So monocytes, macrophages, they are antigen presenting cells. So say that the monocyte, okay, say that a monocyte comes into contact with COVID-19. What it's going to do is that it's going to consume it, right? It might consume it because it might be attached to another cell that might be asking for help, or it might find parts of that. It might. This is exactly how a vaccine works, right? The vaccine is going to inject, if it's the Pfizer or Moderna, they inject you an mRNA. The mRNA then causes you to make a fake spike protein, and then the macrophage eats that fake spike protein and then goes and presents these fake spike protein on the surface of its cell. It is an antigen presenting cell. That spike protein is the antigen, the antigenic part of the virus. So what it does is that it will promote, right, the interaction between that and something called a lymphocyte, which is a type of lymphocyte, to be specific, called the T helper cell. That's a type of lymphocyte. And the T helper cell is the one that goes and activates the second part of your immune system. All right, so we'll talk about that, the lymphocytes. We'll talk about that in just a second. So the macrophages are part of the monocyte, that mononuclear phagocyte system, which includes, write this down, right? Obviously your monocytes, which are the precursors of macrophages, so both of those, but also includes dendritic cells, okay, which are macrophages that are found in your skin, and they are also going to include some lymphocytes, because some lymphocytes are phagocytic, which means that they eat cells. All right, so they are going to eat cells like bacterial cells, or they eat parts of cells that might be dying. They eat anything, right? That's what they do. They're scavengers. That's what macrophages do. And if it's antigenic, then it's going to present it on the surface of its cell. And the marker on the surface of its cell is called the MHC, or major histocompatibility complex type 2, on all of the macrocytes and macrophages and monocytes. And then it's going to have, it's going to motivate other cells that are also can do that, like again, the dendritic cells, okay? So you have, and I'm looking at your, important stuff here in this other site, okay? So the, on the other page, the other type of cell, so these 4 cells are called the non-specific immunity. So it is actually the second line of defense. So write that down. The first line of defense is your skin, your mucous membranes, the product, whatever you have that's called lysozymes in your tears, in your, in your nose, in the liquid, in the fluid, the mucus in your nose. All of these things are your first line of defense. The second line of defense is your non-specific immunity. That's all of the cells I just mentioned, the neutrophils, the eosinophils, the basophils, and the monocytes. These are non-specific. They eat anything that doesn't belong in your body. But then your specific immunity is from your lymphocytes. And your lymphocytes are actually gonna come from a lineage, which means its precursor cells are from the lymphocytic and lymphoblastic cell line. All the other cells come from the myeloid stem cell line. So the other 4 cells come from myeloid stem cell line, and these cells come from the lymphoid stem cell line. So the lymphocytes are going to be your T lymphocytes and your B lymphocytes. And this is really important because they're your specific immunity, but they do specific immunity in 2 different ways. Your B lymphocytes have a type of B lymphocyte that is called a plasma cell, and these are cells that make antibodies. Okay, so antibodies are going to go around, and they are these little Y-shaped things, right? And they go around and they are like a jigsaw puzzle, and they find the things that match up with them, and then they basically try to eliminate them. They tag them, and then they're going to get eliminated by a process called complement. So complement activation sometimes involves a lymphocyte, and the lymphocyte comes because lymphocytes are part of that mononuclear phagocyte system. They eat up that particular cell that's been tagged, okay? Sometimes the macrophage will do that. And so your B cells have a type of cell that's called the plasma cell, and plasma cells make antibodies. There are 5 different kinds of antibodies, all right? So there are 5 different types of antibodies. This is actually part of your signature assignment. Signature assignment talks a little bit about the 5 different types of antibodies, okay? Antibodies are going to be IG, because they're immunoglobulins. It's IgA, IgE, which I already mentioned, IgM, IgG, and IgD. All right, so write these down. I'm gonna go in the order I just said them. IgAs are found in your mucous membranes, like your mouth, your nose, your throat, anything that's moist, okay? They are going to help with like tears, and they're gonna help with things trying to invade your body through your mucous membranes, okay? So that's IgA. It's a particular type of immunoglobulin or antibody that is going to be in your mucous membranes. That's your IgAs. Your IgEs, I already mentioned, are the ones that mediate that allergic response. They're gonna cause anaphylaxis and anaphylactic shock that is what it gets to, right? So IgEs are part of our allergic response, but they are an antibody that's made by a plasma cell, okay? And then we have IgMs and IgGs. So IgMs and IgGs kind of work together. IgM, so write this down, are the primary response. So whenever you are exposed to a virus like COVID-19 for the first time, you're going to make an IgM. An IgM, I call M for immediate, right? So it is the immediate response that your body has. But then the cell, the plasma cell that makes IgMs, once it's instructed and it has been exposed, it converts itself into a plasma cell that makes an IgG. And IgG provides you making lots of antibodies to protect you against that infection again, okay? So IgMs are immediate. Unfortunately, they work, but they don't work fast enough. So when you're first exposed to like measles, chickenpox, or COVID, you're going to have a response. But then you make antibodies. And once you have antibodies, these are IgGs, then you are going to have some type of protection. Okay. And, you know, COVID-19 gets a little complicated because it keeps changing. So the protection is going to diminish over time. All right. But I need you to write this down. IgGs are what confer long-term immunity. All right. So IgGs are, write this down, 75% of all circulating antibodies, right? These are the ones that cross the placental membrane. So when, and are found in breast milk. So when mom has a baby, none of the other antibodies can cross. IgMs, for example, are too big, but IgGs are small. They're only a Y shape, and you'll have to look at the structure because their structures are a little bit different. They are super tiny and they can cross the placental membrane. These are not cells, they are antibodies, okay? They are like a macromolecule. And this macromolecule can cross the membrane because it's super tiny, right? So it confers immunity. It confers immunity to things like, you know, like if you have an MMR, right, which is the measles, mumps, rubella vaccine, the tetanus, diphtheria, and acellular pertussis, the Tdap vaccine, all of these are gonna promote you making an antibody, and then it gives you pretty much long-term immunity. Now, it's not lifelong immunity. Sometimes we have lifelong immunity. It just depends on that particular virus and whether or not this virus mutates, okay? So, for example, measles, mumps, they're not gonna be, you know, these are not viruses that mutate very easily, but COVID, unfortunately, is one that mutates very easily, as you can see by all of the variants that keep coming out. All right, so they are going to mutate. So they confer long-term immunity. So if you ever get your titers checked, TITERS, that's what they're checking for. They're checking for your amount of IgG. And IgG, when passed from the mother to her baby, is called passive immunity, right? So it's passive immunity because the baby did not get exposed and make its own antibody. It is just receiving the antibodies from its mother, okay? So we call that passive, and it's naturally passive immunity, okay? So natural passive immunity. Artificial passive immunity, have you heard of mononuclear antibodies? Yes, mononuclear antibodies, which they give you now for COVID. So what they're doing is they're giving you and injecting you with antibodies, okay? So monoclonal antibodies. They are giving you antibodies into your blood so that it can help you fight COVID. So if you give it, that's called, you know, artificially acquired passive immunity, right? So it's artificial because somebody injected it, you got it from, a company that makes antibodies, but it's going to help you with your immunity. Now, the other types of immunity, there's 4 types of immunity. 2 of them are passive, right? Natural passive immunity and artificial passive immunity. And then the other ones are natural active immunity and artificial active immunity. So natural active immunity is you got COVID, you make an antibody. And then the other is artificially acquired immunity, artificially acquired active immunity is I got a vaccine and I made an antibody so that I don't get COVID, okay? So those are the four: naturally active immunity, natural passive immunity, artificially active immunity, and artificially passive immunity. So you want to make sure you know the four of those. Okay, and so antibodies are really important. By the way, IgDs are not that important, so don't worry about them then in any way, shape, or form. But you do want to make sure that you know those, especially which ones, which type of antibodies are the most abundant, which are IgGs, and which ones cross the placental membrane. Okay, so those are basically part of your immunity that is what we call humoral immunity that involves immunoglobulins or antibodies. That's your B cells. Your T cells are gonna provide what we call cell-mediated immunity. So they don't use an antibody to do their work. The cell itself does the work, okay? So this includes your T helper cell, and then your killer, your natural killer cells, and which are actually part of your non-specific immunity, but also includes the T lymphocyte and T memory cells, okay? So these provide long-term immunity, but cell-mediated immunity, which will go, again, lymphocytes can go and kill, cells in the mononuclear phagocyte system, so that they will phagocytose these types of cells. Okay, so so these two, the antibody-mediated immunity, which is involving B cells and what we call our humoral immunity, and our cell-mediated T lymphocyte T cell immunity, those are both going to be part of our adaptive and or specific immunity. You can call it either adaptive or specific immunity, okay? So that's important for you to know. Now, you'll know, and you'll do a lot more, right, about the immune system actually when you get to microbiology. Because remember, microbiology is going to involve, viruses and bacteria, right? So viruses are usually, right, a piece of either DNA or RNA, and they are not full cells. They are just DNA or RNA. And so they must reproduce inside your cell as a host cell, right? So essentially, they're like parasites, right? They're kind of like parasites. They do not at all live very easily outside of the body, okay? And so if you've ever heard of autoimmune disorders are most often the an incorrect or erroneous response of the immune system to make an antibody against part of your own body, okay? So it's an incorrect, or I call them like oops antibodies, and it's usually antibodies that are made against part of your own body. And so your cells and your antibodies, and then of course the cells who try to kill things that have antibodies attached, like your lymphocytes, they're going to try attacking your own tissues, which is really kind of unfortunate. Okay. Now, let me see, I'm going to look really quick. All of the stem cells that make lymphocytes are going to be located in the bone marrow. That's what the bone marrow is. So let's say that you had an autoimmune disorder. You have rheumatoid arthritis. Rheumatoid arthritis means that you have a plasma cell, a type of B cell in your bone marrow somewhere that is attacking you, that is making an antibody that's attacking the lining of your joints, the synovial lining of your joints, right? So what we can do is we can kill all of your bone marrow, right? We can give you chemotherapy, kill all your bone marrow, give you new bone marrow. I mean, this is obviously very dramatic. Give you new bone marrow, and you should be able to stop or slow down significantly the progression of an autoimmune disorder. Keep that in mind when you're doing your signature assignment, because your signature assignment is part, does talk about an autoimmune disorder. All right. So, and then to get a little bit further into this, and I'm almost done with chapter 24, you want to, so B lymphocytes are made from the bone marrow. T lymphocytes, just another thing you want to write down, they mature in the thymus, right? And so T lymphocytes don't make the same types of errors that your antibodies often do, because the T lymphocytes are instructed on how to attack foreign antigens only. It does, any T lymphocyte that attacks your own antigens, because we have our own antigens, they come from the human leukocyte antigen gene, and they're basically your tissue type, which is actually 6 different things that are part of our major histocompatibility complex. So the gene that provides that major histocompatibility complex is going to be the HLA gene. The major histocompatibility complex is the thing on the surface of the antigen on the surface of our cell that identifies our cells to our immune system as our own cells. There's actually 6 parts of that. Everybody has 6 parts. You get 3 from your mom and 3 from your dad. Okay, so you're never likely to match a tissue type of your parent because your parents were probably not very closely related, but you are more likely to match your sibling, hence the movie My Sister's Keeper, if you watched that movie. Okay, so T lymphocytes mature in the thymus. They don't very easily make these types of oops and cause autoimmune disorders. Autoimmune disorders are made by B lymphocytes that are in the bone marrow that are going to make an erroneous or incorrect antibody. Okay, so one extension of this is our cells everywhere else in our body have human leukocyte antigen, have MHC1 and MHC2, which is predominantly found on your antigen presenting cells. All the other cells have MHC1. It's like a cluster of antigens. But your red blood cells do not have the HLA and MHC like the rest of your cells do. So like if I needed to get you a liver, I need to match your HLA genes with your, right, which make the antigen, which is a protein on the surface of your cell, which is the MHC. I need to match that if I want to get you a liver. But if I wanted to give you blood, red blood cells don't have the HLA MHC. Instead, they have a blood type, right? Either A, B, or O blood type, which is the antigen present on the red blood cell. So if you have the gene for A, then you will express A on the surface of your red blood cells. If you have the gene for B, you will express B on the surface of your red blood cells. If you have A and B, you will express both, and you will have blood type AB. And if you don't have either one of those, you don't have A, you don't have B, then you are considered O blood type because you don't have either one. Okay, O is basically the letter O, which is circle, no antigens on the surface. All right. So if me, for example, I'm blood type B and I'm B positive, right? To be, more specific. And so me, I cannot receive A blood or AB blood because of the A. I am B blood type, and so therefore I have a cell in my bone marrow that makes the IgM that is going to block any type of A blood from entering my body. So as soon as the A is there, my antibody is going to come and bind to it and clump all of the red blood cells that have the A. I cannot receive A blood. If you are A, then you can't receive B blood for the same reason, because you have a B lymphocyte somewhere that's making an IgM. These are IgMs, which are very big, so they don't cross the placental membrane. And so they block, right, your ability to receive any type of B or AB product. So people who are blood type A can only receive A and O. People who are blood type B can only receive B and O. O has nothing on the surface. Remember, it is a blank circle. So they can totally receive. That's why O is the universal donor, right? Because it doesn't have A and it doesn't have B to interact with those people who have A or B with the antibodies that they have, okay? So remember, I am type B, I have antibody for A. And so if you are type A, you have antibody for B. If you are type AB, you have neither. You can't, because you have both of the types. So you have no antibody. So you can take whatever blood. You can take A blood, you can take B blood, you can take O blood, you can take AB blood. AB is what we call the universal recipient. They can receive any blood. O, unfortunately, has both antibodies. Has the antibody for A and it has the antibody for B. So O people can't receive, they cannot receive A and they can't receive B, and obviously they can't receive AB. So they can only receive O. So O people are what we call a universal donor. They can give to everybody, but they can only receive O because they have both antibodies. Remember, antibodies are floating in your plasma, and your red blood cells are red blood cells, the other part of your blood. All right, so you want to make sure you know a little bit about AB blood and the antigen antibody situation with your AB blood typing. All right, so let's go ahead and move on to chapter 26. So chapter 26 talks about reproduction, and reproduction is, very important, obviously, because we are genetically going to be either X, which are females. They are going to be genetically coded to develop in a certain way. And then the XY is going to be a male, because the Y chromosome has in it something called the SRY gene. And the SRY gene produces the testes determining factor, which then is going to cause the development of the production of cells that make testosterone. So testosterone is then going to masculinize the baby, okay? So that's going to masculinize the baby. So in order for a boy to be made, there has to be a Y, which has the SRY gene, which is going to code for testis determining factor, which basically is going to, down the line, start making testosterone. Okay, so the testis determining factor, okay, is the product of the SRY gene, which then it goes and activates and masculinizes, right? The other part of masculinization, because a baby is born as what we call bipotential, it is going to have both male and female parts. If the Y gene is present, we turn on the male parts, which we call the Wolffian ducts, and they have a SRY gene, the testis determining factor makes something called anti-Mullerian hormone, which degenerates the Mullerian system, which is the Mullerian system is the system that would have turned a bipotential embryo into a female. But only if the SRY gene, which is on the Y chromosome, only if the SRY gene is present do you have the testis determining factor that then is going to masculinize the embryo. If the Y gene is not there, again, they get XX chromosome, then they're gonna turn into a female. Okay, if there's no Y, then the default sex is female. Okay, the default sex is female. All right, so a bipotential embryo is going to be masculinized, right, by the testis determining factor, okay, the testis determining factor. So again, the Mullerian ducts are going to be female, and they turn into the Mullerian ducts would turn into the ovaries and the fallopian tubes and the uterus. Or if the SRY gene is present, then the Mullerian ducts would receive something called anti-Mullerian hormone, and they would degenerate, and they would receive instead, they would develop instead the Wolffian ducts, which would turn into testes. And the gubernaculum was the testes actually developed inside by the kidneys, and they're pulled down into the scrotum. Okay, and so they get pulled down into the scrotum by the time the baby's born, right? So the egg and the sperm are gonna carry 23 chromosomes. 22 of them are half of what I have, for example, and then my partner would have the other half, and then I would have one X. All of my eggs have X's because I'm a female, right? The male has half his sperm have X's and half his sperm has Y's, and then fertilization occurs when one of those either X or Y sperm fertilizes an egg, okay? So that is going to be how that occurs. And so the egg has 22 regular chromosomes and 1 sex chromosome, and the sperm has 22 regular chromosomes and 1 sex chromosome, and they each have 23, and when they come together, then you have 46 chromosomes, which is the normal amount of chromosomes that any person should have, okay? There are some errors, and I can talk about them, but then that's gonna take a while, okay? So in the male, right, the testes has a few parts to it that are pretty important. But the main part of what the testes is actually composed of is a seminiferous tubule. So a seminiferous tubule is a tube, and there's a bunch of little tubes inside that testes that are all kind of squiggled all in there, okay? And all these tubes have is a bunch of cells. They have what we call the spermatocyte cells. And you have 2 hormones that are very important that are going to have different effects on the cells of the testes. So they have the spermatocytes, you have Sertoli cells, and you have Leydig cells. Okay, those are the 3 cells that are present in your seminiferous tubule. Now going back to our hormones, do you remember the hormones that I mentioned? FSH, follicle stimulating hormone, and LH, which is luteinizing hormone. FSH and LH, which are released from the anterior pituitary. In a male, the FSH and LH are going to target the testes. As it does in the female it's going to target her gonad, which is the ovary. So the male gonad is the testes, the female gonad is the ovary. So then they're going to, FSH and LH in the male, they're going to target, write this down, right? FSH targets the Sertoli cell, right? And the Sertoli cells provides nutrients for the spermatocyte. So notice they all have S's, FSH targets the Sertoli cell, which might be called the sustentacular cell, which also is an S. And they are going to influence, right? They provide nutrients for the spermatocytes so that the spermatocyte starts to undergo mitosis. Mitosis provides, remember, an exact duplicate. So an exact duplicate, right, with 46 chromosomes can then start to undergo spermatogenesis. Spermatogenesis is going to cause, it's creating the sperm, right, which is going to have 23 chromosomes instead of 46. So they don't do mitosis, they do meiosis. So meiosis is going to occur. And remember, meiosis is one replication of your DNA, but two divisions instead of only one. So you got prophase 1, anaphase 1, metaphase, prophase, metaphase, anaphase, telophase 1, and then you have prophase 2, metaphase 2, anaphase, metaphase, anaphase, and telophase 2. Okay, so there's two divisions. And so you're gonna have replicate the DNA, right? And then you are going to divide once and then divide again. So then you get 4 sperm, each one with 23 chromosomes. All right, that's meiosis. And sperm cells do that very well. So remember, FSH targets the Sertoli cell or sustentacular cell, and that provides nutrients to the spermatocyte, so the spermatocyte can start to, first it undergoes mitosis, and then one of those undergoes spermatogenesis to make sperm. After spermatogenesis, they do spermiogenesis, which is when they take the shape of the sperm, as you know, because you know that the sperm has just a little head with a tail. The only flagella in the human body is found on the sperm cell. So that's called spermiogenesis, the ability for them to actually take that shape. And then once they take that shape, they need to mature. And then so the other part of your testes is the epididymis. That's where sperm matures. And I need you to know this because you're going to be health professionals. It takes 48 hours for sperm to mature. All right. So in your discussions with people who, again, are going to ask you for medical advice, because that's just the way it is that you need to tell people, if you want to get pregnant, you can't have sex 3 times a day because it takes 48 hours for sperm to mature. So have sex once every 3 days to ensure that the sperm is mature, okay? And that first ejaculation is going to be the one that has the most amount of viable sperm. The more the sperm, the more likely you are to have a higher sperm count is going to more likely cause fertilization. Of course, you want to make sure you do it on the right day, but we'll get there because we're talking about the male, not the female yet. Okay, so make sure that they understand that. If you have, ejaculated, the second ejaculate is not going to have as much sperm count, probably not enough to fertilize an egg. I mean, ultimately, it only takes one, but the actual process of getting the sperm there and letting the, processes of the female body eliminate sperm, it makes it very hard, for, if it's not 20 million sperm, for there to be fertilization. And yes, it requires more than 20 million sperm in order for the egg to become fertilized. Okay, so again, that's the part of the male that you want to know. So now, LH in the male, again, LH and FSH are coming from the anterior pituitary. LH targets the Leydig cells. Notice that, right? Isn't that awesome? The Leydig cell, LH, L for Leydig. The Leydig cell produces testosterone. And so testosterone is going to, really what testosterone does is that testosterone creates the male drive for sex, okay? So it causes them to be excited about having sex. So the females also have testosterone, but their testosterone levels come from predominantly from their adrenal cortex. So it's not usually as high as males, unless, of course, they have polycystic ovaries, which is an entirely other conversation, but that does increase the amount of testosterone. Hence, when women have polycystic ovary syndrome, PCOS, they tend to have, you know, sometimes facial hair that starts to grow, chest hair that starts to grow, and other, you know, kind of more masculinizing characteristics, okay? Because they have an increased amount of testosterone. And of course, the sex drive comes with that. All right, so sperm production occurs in the testes, right? It is coming from the spermatocyte, right? FSH is basically gonna encourage the Sertoli cell to secrete nutrients to cause the spermatocytes to divide. The LH is gonna target your Leydig cell, also called the interstitial cell, to release testosterone, which causes the drive. Okay, now in the female can get a little bit more complicated, right? So in the male, the FSH targets one cell and the LH targets a different cell. In the female, you're gonna have two situations that are going, because the FSH and LH are really going to target the cells in your ovary. But the cells in the ovary, right, which is the follicle, they are going to release hormones, which then are going to influence the uterus to prepare the uterus for implantation. So you have your ovarian cycles, which this is the picture that I'm going to give you. I'll send it to you with the recording. And then you have your uterine cycles, okay? So FSH, just like in the male, it targets the sperm cell to mature and to make more sperm. In the female, FSH targets the follicle, which has the egg in it, and that to reach maturity. All right, so in a female, meiosis doesn't really work in the same way as it does in the male. It is, I mean, it does in terms of division, because you do get 23 chromosomes, but the way it works is that it works throughout life and actually is really dependent on fertilization, all right? So oogenesis, the production of eggs, is very different than spermatogenesis. Spermatogenesis is like the embodiment of meiosis. In a female, the egg, we divide and make a lot of eggs, and we're born with the amount of eggs that we are going to have, and they are ready and they're waiting in our ovaries for when they are selected by FSH to be the egg that matures. All right. So once this egg starts to mature, it gets bigger and bigger and bigger. It goes from a primordial follicle to a primary follicle to a secondary follicle to a Graafian follicle. This occurs in the ovary. So write this down. In the ovary, between day 1 and day 14, that's going to be your follicular phase, okay? That is the follicle in the ovary is becoming mature. The bigger and bigger and bigger that follicle gets, okay, the more these follicular cells secrete estrogen. So then the target of estrogen is the uterus. So while the follicle is maturing in your ovary, the egg is releasing more and more and more estrogen, okay? And the estrogen is causing the development of the endometrium. And the endometrium is made up of 2 layers, the stratum basal, the basal layer, the basement layer, and the stratum functionalis. So what it does is that it creates more cells of the stratum basalis. Okay, so the stratum basalis, you get more cells, so it starts to build the endometrium. That's what estrogen does. And that part is called the proliferative phase. And that is part of what's going on in the uterus. So write that down. The proliferative phase occurs in the uterus due to the production of estrogen from the maturing follicle in the ovary. So these are two things that are happening at the same time. The follicular phase in the ovary and the proliferative phase in the uterus. Then the thing that happens that again is a little different than in the male is that estrogen and this increased amount of estrogen is actually blocking the release of LH. It is actually negatively inhibiting your anterior pituitary from secreting LH until your estrogen gets to a certain level. So like, let's say that level is 80%. Once it gets to 80%, bing, then it releases LH, right? Because it gets to a particular amount of estrogen, because the more estrogen that's being produced by the follicle, that means that the egg is now mature. So when it reaches maturity, your LH signal is shut off. And I mean, the inhibition of the LH signal is shut off. And now LH can be released. LH, write this down. The LH surge causes ovulation. So now the egg is mature. LH surge causes ovulation. LH surge causes ovulation. So while in men, FSH and LH are just being like, consistently being sent throughout the month, in females, it's like FSH, LH is being inhibited until it gets to, and then you're releasing estrogen and estrogen and estrogen. When it gets a particular amount of estrogen, then you release LH. So, right, we cycle, right? So then ovulation means the follicle has a bunch of cells on the outside and an egg in the middle. Boom, the egg pops out, right? It's released into the fallopian tubes where it is gonna make this journey as it goes towards the uterus. So let's wait on the journey of the egg for just 1 second and go back to what happens to the cells of the remainder of the follicle. So now these cells that remain in the ovary create this little cyst, okay? And this cyst, instead of producing estrogen, switch to producing progesterone, all right? This little cyst is called the corpus luteum. And the ovulation causes those cells of the follicle, the outside that's not the egg, to convert into a corpus luteum and start to release progesterone and to start the luteal phase. So remember, we're in the ovary. So now the cells that were in the ovary make a little cyst, a little ball of cells that are called the corpus luteum, and they are going to release progesterone. Okay? Progesterone, right, starts to increase, and there's a little bit of estrogen, but really progesterone dominates after day 14 until you end your cycle, right, for 14 days, actually. And so what happens during this luteal phase, again, progesterone is being released, progesterone goes to the endometrium, the lining of your uterus, and causes the development of the stratum functionalis, which is that thick, juicy, nutritious fluid that needs to be there in case that egg gets fertilized, okay? So the luteal phase goes on for 14 days. At the end of the luteal phase, right, the corpus luteum starts to degenerate, stops producing progesterone. Write this down. The drop of progesterone causes then the release of the endometrium. So all of the stratum functionalis is what leaves. The stratum basalis really stays for the most part, but the stratum functionalis is the one that disintegrates, okay? And so then you switch from day 28, if you're a 28-day person, to day 1 and you have menstruation. So the drop in progesterone at the end of the luteal phase initiates the menstrual phase, which is day 1 through day 5-ish. It could be day 1 to day 3, day 1 to day 7, depending on your particular physiology, right? It's a little bit different for everybody. And that resets. And so while you're menstruating, right, FSH is causing another egg to reach maturity. Okay, so that kind of happens at the same time. So your menstrual phase and your follicular phase are kind of happening at the same time. Okay, to the next cycle. So the second half of the, right, in the ovary, you have the luteal phase. In the uterus, you have what we call, oh my goodness. And I've blanked on the name. Okay. Secretory phase. Sorry, I'm like, it's secreting fluid. I couldn't remember the name of it. The secretory or secretory phase, which secretes the fluid that makes the nutritious fluid that's ready for your implantation. All right, so again, when you're not fertilized, you know, the progesterone drops and you are going to cause menstruation. Right? So you go from day 28 back to day 1 and you initiate the menstrual phase. However, okay, remember your menstrual phase is part of your uterine cycle. Follicular phase is happening in your ovary. And then you are going to have what happens if you do fertilize the egg. All right, so remember, fertilization needs to occur. Fertilization needs to occur in basically day 1, in day 14 of your cycle, okay? Ish, very, very much close to the day 14 of the cycle. It's not gonna occur on, it's not going to occur on day 28, okay? That would never happen. Okay, that it just wouldn't happen on day 28. It would have to happen when ovulation occurs. Why? Because the sperm comes in and the egg needs to be like ovulating and day 14, maybe day 13, right before the sperm is there already, or maybe on day 15. But it needs to be around day 14 or the day you ovulate. Because again, if your period is only 21 days, you're gonna be ovulating on a different day. And usually the follicular phase is the short one, not the luteal phase. The luteal phase is pretty much 14 days. And if you're 35 days, again, you're gonna be ovulating on like day 20, not on day 14. The day 14 thing really only works if you're a 28-day cycle. Okay, so then you're going to have, on that day, that's pretty much when the egg needs to be fertilized, because the sperm comes and fertilizes the egg at the very, very beginning in the fimbriae of your fallopian tubes. So then the egg takes about 5 days or so to make its way to the uterus. So then at day 5 is when it can implant. It can't implant really before day 5. So between day 5 and day 7, again, everybody's a little bit different, but between day 5 and day 7, it's reached the stage that it can implant. And if it gets fertilized and it's already in the uterus, because it's, day 22, it's not going to have the 5 days to be able to implant. So it needs really those 5 days of traveling through the fallopian tube, fertilized, dividing, in order for it to be at the right stage for it to implant, right? It's a miracle that all these people get pregnant. It's amazing. So now, when the egg, and this is what you're gonna know, when the egg is fertilized, this is what happens. It releases something called human chorionic gonadotropin, or HCG, which is what you detect on a birth control, on a pregnancy test. Okay. And write this down. This is the important part. This is the clincher. The human chorionic gonadotropin goes back as it's spreading through your body. It goes and makes its way to the ovary, and it keeps the corpus luteum secreting progesterone. What you need, NEED, need to know, is that HCG maintains the corpus luteum so that the corpus luteum continues secreting progesterone, so that, remember, the shut off of progesterone causes the endometrium to shed. You can't shut off progesterone. So the progesterone needs to stay in order for that to create that nutritious fluid for the baby to attach in that uterine lining. So anything that blocks progesterone, because that's what the abortion pill is, which is called RU486. I'm not talking about Plan B. RU486 is the abortion pill that you take that blocks progesterone and causes, boom, the shedding of your lining, okay? And that's the abortion pill, which for a long time it was not approved in the United States, but it has in some states been approved, and that can block progesterone, and it causes anti-progestin, so it blocks progesterone and causes shedding of the lining. Okay, so that's what you need to know. That's the clincher. HCG prevents the progesterone from dropping. The placenta eventually will be able to take over in providing the progesterone. And that handoff usually happens somewhere between the first trimester and the second. So sometimes that handoff between the first trimester and the second doesn't occur. So something happens, the ovaries stop producing progesterone, the female will have a miscarriage. Because again, the uterus shed. So this is why, like, if you make it past the first trimester into the second, that means that handoff between the ovary providing the progesterone and the placenta providing the progesterone was, occurred the way that it should. Okay, so I'm going to go ahead and stop there. I think I covered the majority of it. Again, I'm going to give you a picture that is going to help you kind of see all of these things that I just mentioned. Okay, so thank you so much, and I'm kind of done with chapter 23 and 24.